All right, everyone. Welcome to the third lecture in Chapter 18, Blood. This lecture is going to cover erythrocyte disorders and blood typing. I believe I have the erythrocyte disorders as self-study, but I do want to go ahead and discuss them a little bit because of, if nothing else, because of something that I said in the previous lecture that I think, if I remember right, I actually said it backwards. So we're going to talk about disorders and blood typing. So to begin with, let's do our not really attendance questions. And again, I ask you try to answer these questions before I give you the answers. So try to pause your video and try to answer these questions before listening. That way it'll give you an idea of, are you up to speed? Are you keeping the information in your head the way you need to? So what is the ultimate fate of globin and one molecule of HB, which is hemoglobin, can bind how many oxygen molecules? And go ahead and pause your video and try to answer those, and then we'll see what the correct answer should be. So, what is the ultimate fate of globin? Globin, remember, is the protein portion of the hemoglobin molecule. And when an erythrocyte has served its life and it's being destroyed, that hemoglobin is going to be broken down into heme and globin, and the globin, the protein portion, is broken down even further. And like all proteins, it's made of amino acids. So the globin is broken down into amino acids, which are then recycled to make more proteins. Now, it doesn't have to be more globin. It could be any protein, because they all use those same amino acids. Now, one molecule of hemoglobin can bind how many oxygen molecules? Well, this actually talks about the other part of hemoglobin, the heme pigment. And remember, one hemoglobin molecule has 4 heme pigments, each of which has an iron atom in the middle, each of which can bind 1 oxygen molecule. So one hemoglobin can bind 4 oxygen molecules. So let's talk about erythrocyte disorders. First is polycythemia. Polycythemia is when you have an excess hematocrit, or an elevated red blood cell count. When your red blood cell count is high, that's called polycythemia, and typically this is the result of a cancer of a specific set of cells in your red bone marrow, those cells that we saw last time that lead to red blood cells, erythrocytes. So high hematocrit, high red blood cell count is polycythemia. The other is anemia. Now, in lab, we use anemia just to mean low red blood cells, but really anemia, there's a lot of different types. So anemia is not a disorder. Anemia is a class of disorder, and there's a lot of different types of anemia. So let's talk about some of those. First is hemorrhagic anemia. And what we will see here is all anemias have one thing in common, and that is an inability to carry oxygen because something is going on with your erythrocytes. In hemorrhagic anemia, this emerges because someone is hemorrhaging, losing blood. Well, if you start to lose a lot of blood, then you don't have enough erythrocytes to adequately carry oxygen. It can lead to hemorrhagic anemia. Iron deficiency anemia. In iron deficiency anemia, that's just what it sounds like. If you don't have enough iron, then you're not going to have enough hemoglobin with iron in it, and you can't adequately carry oxygen if you don't have enough iron. Now, pernicious anemia, this is something that I talked about in the previous lecture that I haven't gone back to listen, but the more I thought about it, I think I actually said it backwards. In pernicious anemia, it's an autoimmune disease, and autoimmune disease just means that your own immune system thinks that some part of your body is foreign, so it starts attacking it. Those were mentioned briefly in Bio 137. In pernicious anemia, your immune system thinks that your stomach lining is foreign, so it starts to break your stomach lining down. Now, your stomach lining makes something called intrinsic factor, and intrinsic factor is needed to absorb vitamin B12 and folic acid. Intrinsic factor is made by your stomach lining, and it's needed to absorb B12. Now, B12 is needed to adequately make red blood cells. And if you don't have enough B12, you will be low in red blood cell count. And oddly enough, even though this is typically an autoimmune disease, we're starting to see it more commonly in people who have had weight loss surgery. So let's think about how weight loss surgery works. There's a bunch of different types, but it's really, really common to either remove a part of the stomach or staple off a part of the stomach. You're making a part of the stomach unavailable, which includes the stomach lining. So if you don't have access to a lot of your stomach lining, then you're not going to make as much intrinsic factor. And that can lead to low B12. It can lead to a type of anemia, pernicious anemia. So it's often the case that if someone has had weight loss surgery, they end up getting something like B12 shots to account for that. So usually pernicious anemia is an autoimmune disease, but we will also see it in people who have had weight loss surgery pretty often. Next is hypoplastic anemia, and I want to go ahead and talk about aplastic anemia at the same time because they're really, really similar. Hypoplastic anemia is just reduced function of the red bone marrow to make erythrocytes. So the erythropoiesis slows down a lot. In aplastic anemia, it goes even further and it stops completely. And the reason that it stops completely is something has destroyed the red bone marrow. So hypoplastic, the red bone marrow is not functioning enough. In aplastic anemia, it's not functioning at all because something has destroyed it. And these are really, really common in someone who's getting something like radiation therapy or certain types of chemotherapy because the red bone marrow is really easily destroyed by those. In hemolytic anemia, this is something has caused the red blood cells to rupture. Hemolytic, that literally means breaking apart the blood. LYT or LYS means to break or rupture. So hemolytic anemia, the red blood cells are being destroyed. Renal anemia is when you have some sort of kidney disorder. We will actually see this a lot on people on dialysis, but if your kidneys are not functioning correctly, oftentimes they will not make EPO, erythropoietin. And we saw last time that's what stimulates erythropoiesis. Thalassemia and sickle cell anemia. These are actually both gene mutations that cause the hemoglobin to be shaped incorrectly. And remember from Bio 137, proteins, it is all about shape. And that's going to be really important all semester. Proteins do their job because of their shape. And if something causes them to change shape, such as a gene mutation, then they can't do that job anymore. Thalassemia is common in people of Mediterranean descent. Sickle cell anemia is common in people of African descent, although anyone can get any of these. But thalassemia more common in Mediterranean, sickle cell anemia more common in African, and the result is very similar. In thalassemia, the red blood cells kind of resemble almost like a bullseye under the microscope. In sickle cell anemia, the red blood cells are hooked like a sickle, and they can't adequately carry oxygen. Especially in sickle cell anemia, not only can they not carry oxygen as well, they have a hard time bending and squeezing to get through the very small capillaries. So it can be very, very painful to someone with sickle cell. Now we're going to talk about blood groups, and I'm actually going to present this with a draw along. I do have the chart here that's, I don't think this is the same chart that's in your textbook. There's a similar chart on page 683, but I'm going to do kind of a draw along so that we can see exactly how blood types are determined. So first I want to talk about some terms that we're going to be using a lot, and the first is antigen. Antigens are proteins and in some cases carbohydrate protein combinations that are found on the surface of every cell in your body. But in this chapter we are specifically talking about very, very specific antigens found on the surface of your erythrocytes, on your red blood cells. And we're also going to talk about antibodies. Remember gamma globulins for the first lecture. Antibodies are defensive proteins found in your blood plasma. So antigens are on your erythrocytes. Antibodies are floating in your plasma. Now, antigens are really how we know what blood type you have. So let's look at someone with type A blood. And someone with type A blood, this is what we would see if we were able to look at their red blood cells and their plasma really, really closely. So over here on the left, what I've done is given kind of a key. I'm going to use this thing like a circle on a stalk to represent the A antigen, and triangle on a stalk to represent B antigen. Someone with type A blood will have the A antigen all over the surface of their erythrocytes. Now, remember, plasma antibodies are looking for things that don't belong there. And antibodies, they come in a bunch of different shapes, but typically they're like an upside down Y or something like that. They've got branches. And on the end of each branch is going to be a shape that's looking for something that shouldn't be there. So this would be an anti-A antibody. But we don't have those in someone with type A blood. What we actually see in someone with type A blood are anti-B antibodies. Oh, let me get this shaped correctly. So antibodies are shaped to fit whatever it is that they're looking for. Remember, the anti-B antibody over here is a triangle. So these little receptors on the ends of the antibody branches are going to be shaped like a triangle. Antigen A in someone with type A have antibody, let's go ahead and write this, anti-B. Now we want to say anti in front of it because saying the B antibody isn't really correct. It's the anti-B antibody because it's against the B antigen. Someone with type A blood should never have B antigens in their body. So the antibodies are looking for things that don't belong, and they do that by being shaped in such a way that they would fit against it like a puzzle piece. So let's look at another example. If someone, let's say, has type B blood, then what they would have is the B antigen. And they should never have the A antigen, so they will have anti-A antibodies shaped in such a way that they would fit those A antigens if they showed up. So type B blood has the B antigen and the anti-A antibody. So what you're seeing is the antigen is the blood type. The antibody is the opposite of the blood type. Whatever antigen they have, that is their blood type. But what if they actually had both A and B antigens? What antibody would they have? Well, they shouldn't have anti-A, because A antigens would normally occur in their body. But they also wouldn't have anti-B, because anti-B would cause problems. They should have the B antigen in their body. So their blood type would actually be type AB. Someone with blood type AB, it doesn't mean they have an AB antigen. There's no such thing. Someone with blood type AB, that means they have A antigens and B antigens. But they would have no antibodies because nothing would look out of place. What if we looked at someone's blood, though, and we saw they didn't have any antibodies? What if we looked and their erythrocyte had nothing? Well, that would be they have 0 antigens. So what we did was we called those blood type 0. But when that was happening, we had been talking about blood type A and B and AB, all these letters. Somehow it got lost in the shuffle. Instead of being called blood type 0, we just made that into a letter and called it blood type O. So if you hear someone called blood type O, that just means they don't have A or B antigens. But if they don't have A or B antigens, there should never be A antigens or B antigens in their body. What kind of antibodies would they have? Well, they would have anti-A, and they would have anti-B. They should never have A antigens, so we've got anti-A antibodies looking for them. They should never have B antigens, so we've got anti-B antibodies looking for them. Someone with blood type O has no antigens, but they have both anti-A antibodies and anti-B antibodies. Okay, so let's go back to our PowerPoint for just a moment, and we'll come back. Here's that kind of in chart form. But there is something else that we often hear. Someone doesn't usually say they have A blood. They'll say they have A positive or B positive or A negative or O positive. So what's this positive and negative? This is something called Rh factor. And Rh factor really is just another antigen. It's actually a group of antigens, but usually when we're talking about Rh factor, we're talking about a specific antigen, sometimes called the D antigen. I will instead just call it Rh factor or Rh antigen. And it's a little bit different from the ABO blood group. So let's go back to our drawing and see exactly what it is that Rh does differently and what it does the same. So I'm going to add another item to our key over here and just say square on a stalk is the Rh factor or the Rh antigen. Let's erase our antibody so that we can do an example or two. If someone has circle and square, look over here, we see circle is A antigen, square is Rh antigen. So based on what we've said, it sounds like they should be called blood type ARH, but they're not. We use different terminology. They are still blood type A, they have the A antigen. When someone has the Rh antigen, they are positive for it. So blood type A positive. The A tells us they have the A antigen. The positive tells us they have the Rh antigen. What antibody would they have? Well, if we checked their plasma, they should never have the B antigen, so they will have anti-B antibody. Someone with blood type A positive has the A antigen and the Rh antigen. They are positive for having the Rh antigen. And they should never have B antigen, so what they do have is the anti-B antibody. And that's from the time they're born, they have those antigens, they have those antibodies already in place. Let's do another. Let's say we check and this person has the B antigen and the Rh antigen. What type are they? See if you can answer that. They would be type B positive. They have the B antigen and they have the Rh antigen. So they are positive for Rh. They are type B positive. They have the B and Rh antigen. What antibodies would they have? Well, they would be born with anti-A antibodies because they should never have A antigens. Someone with blood type B positive has the B antigen and the Rh antigen, and they have anti-A antibodies. And they will be born with that, and they will have that their entire life. But let's look at the one time that it gets a little bit tricky. Let's say we look at someone and they have the A antigen but nothing else. Well, if we're talking about the ABO blood group and the Rh blood group, this person would be B. I apologize for that. They have the B antigen, so they are blood type B. Do they have the Rh factor? No. They are negative for Rh. The negative is not another antigen. It means they do not have the Rh antigen. Positive means they do have the Rh antigen. Negative means they do not have the Rh antigen. So what antibodies would they have? This is where things get a little bit different from what we've been saying. They are born with anti-A antibodies because they should never have A antibodies. A antigens. Someone with blood type B should never have A antigens, so they have anti-A antibodies. They should also never have Rh antigens, so it seems like they should have anti-Rh antibodies. but they're not born with them. And they could potentially live their entire life and never have anti-Rh antibodies. But if they ever come into contact with Rh positive blood, from that point forward, they will start to make anti-Rh antibodies. I'm going to put a star next to that, because when we are born and we look at the ABO blood group, we have whatever antigens and the opposite antibodies from the time we're born. But when we're talking about Rh, if we're not born with the Rh antigen, it seems like we should be born with the anti-Rh antibody, but we're not. We don't start making anti-Rh antibodies unless we come into some contact with Rh positive blood. We say, if stimulated. So let me give you an example of that. Little boys, I don't know if they still do this, but they used to do this thing called blood brothers, and you've probably heard of that. They'll like cut their hand or their thumb or something and smear their blood together. I mean, stupidness aside, let's look at what that could happen. If one boy is Rh negative, it would look just like what we see here. They have, let's say one boy is A positive and one boy is B negative. Well, the B negative is what we see right here. They would have the B antigen, they would have anti-A antibody, but they would not yet have anti-Rh antigen. But if the Rh positive boy's blood now comes into contact with him, he has been exposed to the Rh antigen. And he will start to make anti-Rh antibodies, and he will then have those anti-Rh antibodies for the rest of his life. Not really a big deal in that case. But when it becomes a big deal, let's go back to our PowerPoint. It becomes a big deal if we're talking pregnancy. So let's say an Rh negative mother has a child and the father is Rh positive. Well, if this is her first child, it's really not an issue. If the father is Rh positive, the baby could potentially be Rh positive. And mom, when she is carrying a developing baby, her antibodies cross the placenta and circulate in this developing baby to protect the baby. It's kind of a defensive mechanism because the baby doesn't start making their own antibodies just yet. Well, if mom has never come into contact with Rh positive blood and mom is Rh negative, she would not have any anti-Rh antibodies. There's no problem. But when this baby is born, that's kind of a traumatic experience. There's some tearing, there's some bleeding, and mom will then be exposed to this baby's blood. And when she is exposed to the baby's blood, she's exposed to the baby's Rh antigen. And from that point forward, mom is going to start to make anti-Rh antibodies. Not a problem for that baby that she just had. But if she has another baby, the second baby or any baby after that, if that baby is Rh positive, then that baby has Rh antigens on his erythrocytes. And like we saw, mom's antibodies cross the placenta and circulate in the baby. But if mom's now carrying anti-Rh antibodies, if those antibodies are circulating in baby, those anti-Rh antibodies are going to recognize the baby's Rh antigen as foreign. So let's clear this canvas and look at what happens in that case. I'm going to draw some antibodies here. And we can see they're branched. And we saw on an erythrocyte, those antigens, if they are Rh, then they fit together like a puzzle piece. And there would actually be many of those antigens. And there would be lots of antibodies. And more and they would just keep sticking together. And what we can see is that starts causing clumping or agglutination. And the more antibodies there are, the more agglutination. So the baby's blood starts to clump. And further still, once we have clumping, the red blood cells start to be broken apart. We'll learn how that happens later in the semester. It's not the antibodies that do that. I see a mistake that I've done here. There shouldn't be. The baby's blood starts breaking apart. And that's because mom's antibodies that are circulating in baby recognize baby's antigens as out of place. So when an Rh negative mother has had an Rh positive child, that child, the first child's not at risk, but the second child and every child after that, if they are Rh positive, they're at risk for what we just saw. And that's called erythroblastosis fetalis, or hemolytic disease of the newborn. Those phrases are interchangeable. And remember, hemolytic, breaking apart the blood, ripping apart the erythrocytes. And that's because the antibodies from mom recognized the antigens from baby as foreign. Now, this is only a problem if mom is Rh negative, and it's only a problem when her baby is the second or later Rh positive child. So what do? They're going to give something called RhoGAM. Now we're going to say this two different ways. We're going to say this the way just to explain it, but then I'm going to tell you the way that it's actually done. RhoGAM is a drug that suppresses mom's antibodies. So if mom shows up and says, this is my second child, my previous child was Rh positive, I need to get some RhoGAM. They're going to give her RhoGAM so that her blood does not attack the baby's blood. That's the easy, clear way to talk about it. That's the way we'll talk about it in this class. But in reality, the doctor, you remember, if you ever find out you're pregnant, you go to the doctor right away. If you're a woman. I definitely go to the doctor right away if you're a guy and you find out you're pregnant. But no, if you're pregnant, they always say go to the doctor as soon after as you know. And one of the things that they're going to do is do a blood test. And in reality, if mom is Rh negative, they're going to give her RhoGAM. They're not going to ask her, have you had an Rh positive baby before? No, they don't want to risk anything. They're not going to rely on mom's medical history to be accurate. They don't know what she has done before. So in reality, if mom is Rh negative, she's getting RhoGAM. And that's to protect the baby. But for purposes of this class, we talk about RhoGAM when mom is Rh negative and she has already had an Rh positive baby previously, and now she's on her second or later baby. Okay, so to tie everything up, we've talked about blood groups. Let's talk about transfusions, when someone is giving or receiving blood. Who can donate to who? Well, what it comes down to is never introduce a foreign antigen. You can put any antigens at all into a person as long as they already have them, but never put in a foreign or new antigen. Never introduce a foreign or new antigen. So who can donate to whom? I'm not going to go through all of them, but someone who is blood type A negative, that just means A is the only antigen they have. So they can donate to A negative, A positive, AB negative, AB positive, because in all of those cases, we are only introducing A antigens, which is something that that person already has. O negative doesn't have any antigens at all. O negative we call the universal donor, because we can introduce that to anything without worrying about introducing a foreign antigen. AB positive can only donate to AB positive because they have A and B and Rh factor. So they can only donate to someone who has A and B and Rh. So that leaves them only able to donate to AB positive. But AB positive can receive from any blood type. You wouldn't be introducing anything foreign. If you have AB positive blood, you're in luck. You can get whatever blood you want. So what happens if you accidentally introduce the wrong blood type? That's what we see over here. This is called agglutination. So agglutination occurs when an antigen and an antibody come together. If you introduce a foreign antigen, then you're at risk for agglutination and ultimately hemolysis, breaking apart of the blood. Now, there's also the lab video for figuring out blood type, and there we saw agglutination in some fake blood. If you haven't watched that video yet, make sure to. But this is real blood, what would happen. Here's agglutination, agglutination, agglutination. On the horizon now, I've had this slide for several semesters now, and I keep it as on the horizon, but in reality, this is a real thing now. It's not widespread, it's just starting to roll out, but it's really, really interesting. The University of British Columbia has designed an enzyme that cleaves the antigens off the surface of erythrocytes. So if I have a bag of blood of any sort, I can put this enzyme down inside of it, and it will cut all of the antigens off of those erythrocytes. So let's use that as a thinking cap question to wrap up today. Why would that be important? Why would we want to cleave the antigens off of erythrocytes? See if you can answer that before I give you the answer. Well, by cleaving the antigens off the surface of erythrocytes, what have we really done? We've turned that blood, regardless of what blood type it was, we've turned it into type O negative. Remember, erythrocytes with no antigens on the surface, they're not A, they're not B, they're not Rh positive. So they are type O negative. And we just saw that type O negative can give to anyone. It is the universal donor. if you're short on blood or if you don't have time to figure out someone's blood type, you can take this enzyme, put it down in a bag, create O negative blood, and it's safe to give to anybody. All right, so that's where I'm going to end this lecture, and I will talk to you next time.